The overall goal of this procedure is to generate targeted gene-disrupted Streptococcus mutant strains using a two-step fusion PCR and an electroporation.
This method can help answer key questions in the microbiology field, such as how a gene of interest works. The main advantage of this technique is that no enzymatic reaction other than PCR is required, and competent cell preparation for electroporation is quite simple.
Though this method can provide insight into Streptococcus mutans gene disruption technique, it can also be applied to other microbial species such as Staphylococcus aureus.
Generally, individuals new to this method will struggle because they are not familiar with the effectiveness of nested primers for the second PCR.
Demonstrating the procedure will be Dr. Ayako Okada, a research instructor from our laboratory.
To begin this procedure, streak stock S mutans UA159 onto a brain-heart infusion or BHI agar plate and incubate the plate overnight at 37 degrees Celsius under anaerobic conditions. On the following day, use a sterilized toothpick to pick a single colony and inoculate it into 5 milliliters of BHI broth. Incubate the S mutans culture overnight at 37 degrees Celsius under anaerobic conditions. Next, centrifuge the culture at 2000 times G for 15 minutes. Remove the supernatant and resuspend.